Welcome to Spring LIB.
To answer this question, we have to understand what the central dogma is and figure out which components of the central dogma are going to most represent a disposable photocopy. All right, so our central dogma starts with DNA, where we transcribe it to produce RNA, and specifically it's mRNA that is translated into protein. All right, so going back to our answer choices, right, we have DNA, protein. Well, looking at our central dogma here, which are most like a photocopy? Well, the DNA is not a photocopy. You can't just get rid of it, because if you get rid of the DNA, well, none of this can occur. So that's not right. What if we get rid of the protein? Well, that's kind of like you did all this work. You went through all these steps to finally produce the protein. Now you're going to get rid of it? Well, kind of seems counterintuitive. So it's not necessarily like a book that's a photocopy, right? So we know it's not DNA, it's not protein. What about tRNA? Well, tRNA is not going to be a photocopy either, because it, although is kind of replaceable, the tRNA, its job is to carry the amino acids to the ribosome when they're needed, specific amino acids. So it wouldn't be tRNA. What about the mRNA? Well, the mRNA is like the photocopy, because it is essentially a transcript. And transcribe, when you say transcribe, it basically means to take something and copy it down. So when we say transcription, we can call it copying. So the mRNA is basically a copy of DNA, except the mRNA is going to be single-stranded, and the thymines, the T's of the DNA, are going to be exchanged for uracils, U's on the mRNA. So that's the main difference between the two. And of course, one is ribonucleic, mRNA, the other one is deoxyribonucleic, and that's the DNA. But other than that, it's basically a copy of the DNA. So which would be a disposable photocopy? Well, it would be the mRNA. So our answer here is answer choice B, mRNA.